About my research today, which focuses on patterns and processes of evolution, and the processes I'm most interested in are speciation and extinction. So if we had the complete history of a group of organisms, we'd have something that looks like this, where we have not only the extant species that survive all the way to the present, but also these red dotted lineages, these that have gone extinct, and where we have a fossil record. But usually what we really have is something more like this, where we have just the phylogeny of living species. And so I'm really interested in whether we can tell things about the processes of evolution from the patterns in these extant phylogenies. So there's been this is something that paleontologists have been doing for a very long time, but it's also become something that molecular phylogeneticists have been working on for the last few decades, with a lot of papers coming out that are trying to figure out how we can get at speciation and extinction from extant phylogenies. And some showing and among others, Dan Roboski here, who has shown that speciation and extinction rates changing over time can really make it difficult to tease these apart. And we really have to try to find ways to parameterize change through time. Also, there's been a lot to show that the diversification dynamics we see from the fossil record are often incongruent with the patterns we get from the extant phylogenies. And so there's been new methods to try to reconcile these differences. The main difficulty that most have been proposing is that extinction is really hard to estimate from these phylogenies, and perhaps we shouldn't even be trying to estimate extinction, while others have suggested, yes, maybe we can, and then some backlash to say, no, we really can't. So some of the latest methods have really tried to parameterize everything possible, so changes in rates of speciation and extinction, not only through time, but also across the tree, and that this might be able to accommodate some of these difficulties in estimation techniques. So I'm interested in testing these questions with the order Primates. It's a diverse group where the extant species have a wide range of form and function, and they're distributed across the global tropics. But the diversity that we see today really pales in comparison to the diversity that once existed. The primates are a great group to use as an empirical system because they have an exceptional fossil record. We have some of the oldest primates from 50 to 60 million years ago, as well as those fossils that are on the lineage leading to humans. And so primates also vary in their diversity, for example, across the globe. So we have monophyletic lineages in the New World, and we can compare those to the Old World. There's variation in clade ages, where some of these clades have similar ages, and we can test if time has an effect on diversity. But again, that diversity that we see today, about 400 species, is really just a shallow snapshot of the diversity that once existed. And so you can see there's more than 700 species that have gone extinct. Furthermore, these species lived in places where primates no longer live today. For example, North America, Europe, some of these desert zones. So again, lots of opportunities to test the causes of variation in diversity. So I started by assembling a database of primate alpha taxonomy in the fossil record, as well as the first and last appearance dates, or their ages, and records of their occurrences. And I started by just downloading the Paleobiology database and then vetting that database against about 90 primary sources to get the dates to be as precise as possible, to fix up the alpha taxonomy to be as accurate as possible and as up to date. And this is just a breakdown of how the number of species fall into these different taxonomic groups, where the adapids and plesiadapids are entirely extinct stem lineages, and the tarsiers are now only represented by about 10 living species, but they were once far more diverse. The lemurs and their relatives, the strepsirrhines, New World monkeys, and I really want to point out the Old World monkeys, which is the biggest group. And so in total, there's about 1,100 primates. This number actually kind of shocked me because I really only knew about the 400 that are alive today. But this 700 plus fossils really represents the majority of the diversity. And then I created a kind of super tree where I merged the most complete phylogeny for the haplorhines, or the tarsiers, monkeys, and apes, with the most complete phylogeny for the strepsirrhines, or the lemurs and lorises. And this is a breakdown of that tree at the genus level to show with the bars across the tips representing the proportion of sampled species. And you can see that for most of the genera, we have 100% sampling. But I can also correct for this non-random missing species across the tree by assigning the sampling fraction per genus. But you can see that overall it's a pretty complete tree. And this is just color coding the tree by the number of species in each genus. So you can see that most genera have fairly few species, while a few genera stand out as having very high diversity, such as this group of lemurs, a group of New World monkeys, and a few of the Old World monkeys. To analyze the fossil data, I used a birth-death process in a Bayesian framework, which jointly estimates speciation, extinction, and preservation rates, as well as shifts in those rates through time. And I analyzed the data with all the primates together, as well as splitting up different taxonomic groups separately. And just to pause for a second to discuss preservation, because that is a big deal with the fossil record, in this model, preservation is modeled as a non-homogeneous Poisson process, where if we were to describe the probability distribution, we call it like a hat-shaped distribution, where the probability of sampling fossils is lowest at the earliest beginnings of speciation for a species and lowest towards the end of its lifespan, and the probability of sampling is greatest kind of in the middle, where the abundance of the species is probably highest. This is kind of a natural way of modeling that preservation bias. This is just one of the main cruxes in this model, and I kind of wanted to point it out to show you where the data really parameterize the model. So here, the probability of the speciation and extinction times, given the speciation and extinction rates, is a result of the speciation rate raised to the number of speciation events, and the extinction rate raised to the number of extinction events, and it's also parameterizing the sum of species lifespans. So this is how those fossil data really calibrate the model. And then to analyze the phylogeny, I used a method of reversible jump Markov chain Monte Carlo that jointly estimates speciation and extinction, as well as shifts in those rates through time and across the tree. This Bayesian analysis of macroevolutionary mixtures, or BAM. And just to give you one of the main parts of this model, the instantaneous speciation rate is a result of the initial speciation rate and this exponential function, which is a natural way of trying to model change through time with this rate change parameter. So if there's no evidence for a change in the rates through time, this parameter just goes to 0. So to jump right into the results, here is a lineage through time plot from the living phylogeny. And so you can see that on this log scale, you have pretty much this linear increase in the number of species through time. Contrast this with the fossil record, and we can see that there's more of this waxing and waning diversity pattern through time. And especially I want to point out how early in the primate divergence, we have a lot of diversity that's not picked up by the phylogeny, and this is those stem lineages, which we simply can't model with the extant phylogeny. And then I really want to point out this time period, which is the Oligocene, and this is a time period of known global cooling and drying, which is thought to be a period of mass extinction for most mammals, especially primates. And yeah, primates did take a pretty big hit, but there was a lag here, and I'm really interested in exploring more why there is this lag before the diversity really takes a big hit and then rebounds into the Miocene. And then I also want to point out this uptick in diversity towards the present. We're going to talk more about that in just a moment. So from the BAM analysis, the diversification rate as estimated from the extant phylogeny suggests that diversification has increased through time and has been highest towards the recent. And this is driven mostly by speciation rate increasing through time, and extinction rate kind of tracks speciation throughout most of this history, but it's much lower than speciation, leading to this net positive diversification. Contrast this with the fossil record, and what we see is more or less a 0 net diversification through time. And this is driven by speciation and extinction rates actually increasing, but they pretty much track each other. There's almost this constant turnover of species through time, and leading to this overall pretty much net 0 diversification. I also like to point out that the speciation and extinction rate estimates differ by almost an order of magnitude between what we get from the extant phylogeny and from the fossil record. And then just to break it down a little bit further by taxonomic group, we can see that this, from the extant phylogeny, this inference of increasing diversification rate is really driven by one clade, the catarrhines, or the old world monkeys and apes. Whereas from the fossil record, we can see that most of the taxonomic groups have had a more or less 0 diversification rate until fairly recently, where some of these taxonomic groups have had markedly negative diversification rates. The BAM analysis found evidence for 2 shifts in diversification from the extant phylogeny, one at the node for the anthropoids, which is the monkeys and apes, and another one for the old world monkeys and apes. And so here the tree is color-coded by the instantaneous diversification rate estimate. So the red is 0.5 is the highest diversification, and blue is the lowest. And so this result is driven by, from the old world monkeys and apes, this estimate of an increasing speciation rate through time, with a decreasing extinction rate through time. Again, contrast this with the fossil record, and the old world monkeys and apes seem to have exhibited almost constant speciation and extinction through time, with this little bit of a blip towards lower speciation, and that results in this almost net zero diversification through time. So to summarize, I found incongruent patterns of diversity and diversification between the fossil record and the extant phylogeny, where the fossils showed this kind of waxing and waning diversity pattern through time. The phylogeny suggested this exponential increase in diversity, whereas the fossils suggested zero net diversification, the phylogeny suggested increasing diversification. This was caused by incongruent patterns of speciation and extinction rate estimates. So from the fossils, we see that speciation and extinction kind of tracked each other through time, this turnover of species over time, whereas from the phylogeny, we get divergent patterns of speciation and extinction. This may suggest that our interpretations of diversification dynamics may be limited from extant phylogenies, or we maybe have to just isolate our interpretations to the extant taxa only. But hopefully we'll hear in the next talk about ways that we can resolve these incongruences. In the next steps for my research, I'm interested in correlating patterns of diversity and diversification through time with external environmental variables, such as the global temperature estimates for the Cenozoic, as well as intrinsic traits of the species, such as their body mass estimates. And body mass has changed markedly through time, not only the mean estimate, but also the variation in body mass. And this is all based on the fossil record. So that's where I'd like to take the research next. And that's all I have. I'd like to thank the museum for their funding, my supervisors. I'd especially like to thank the New York Consortium for Evolutionary Primatology for really helpful discussions on this research, Liliana Davalos, and to thank you all for your attention.
Fossil-bearing rocks for mammals through time, and if that varied at all, like you have that dip in primate diversity at the Oligocene. I just wonder if there's also fewer possible rocks that would have primates at that time.
Yeah, that's a great point. It's basically the preservation problem. Is it a matter of we see extinction at the Oligocene, or is it simply that we have a preservation bias? And both are possible. The only thing I can really say is that the analysis kind of takes that preservation rate change through time into account. But we do see this same pattern in other land mammal fossil assemblages where during the Oligocene there is a big dip in diversity. But I think that is something that we would want to look at continent by continent perhaps, and that might help to understand if preservation is biased by land masses as you suggested.
Yeah. How did you determine whether or not a particular fossil taxon was a valid one? Because you didn't have time, I expect, to personally examine every single fossil taxon known.
Yeah, so just in case you didn't hear, the question is basically about the alpha taxonomy and how do I know it's right. The only thing I can really say is that I took a splitter's approach to both the fossil record and the extant phylogeny.
So any name that was in the literature you accepted? Within
reason. Any name within reason. So there is debate over particular genera and particular families. And so I reviewed the literature to the best of my ability. Where I felt that certain things were being completely oversplit, I erred on the side of caution. But yeah, I think one way to address that is to have one analysis like this, which is the splitter's taxonomy, and maybe one with a more lumpers taxonomy.
Or you could accept one particular source for that information.
That's another way. Thank you. Yeah. Thank you.